Reproduction and embryonic development. Before we talk about reproduction, let's talk a little bit about sexually transmitted diseases. These are diseases transmitted by the process of having sex. STDs can be caused by bacteria, protists, fungi, and yes, viruses. And that's where the problem is. Viruses are tricky little bugs. They can hide inside cells, they can mutate, and it makes it very difficult for the immune system to catch up and eradicate them. Typically, once you're infected by a virus, especially the viruses that cause sexual diseases like genital herpes or genital warts, I might add, once you catch the virus, it's with you the rest of your life. The best prevention is the key to prevent any sexually transmitted disease. And these are actually photos of actual viruses. In chapter 27, we got quite a bit to cover. We're going to look at asexual reproduction without sex and sexual reproduction, human reproduction, the principles behind that, development, the basic principles of embryonic development, which are shared amongst different groups of animals with an emphasis on human development. Reproduction maintains the species. Although an individual may have a short life frame, his or her genes can live on in the offspring. Reproduction is simply defined as the creation of new individuals from pre-existing ones. Animals reproduce in one of two fashions: sexually, which involves eggs and sperm, or asexually. Asexual reproduction only involves one parent and production of lots of offspring in a short period of time. Within the animal kingdom, reproduction can be by way of budding, where the new individual grows from the parent. Parent is still conserved. Fission, we see this in protists, where the parent splits into two or more offspring about equal size. Or fragmentation, regeneration, where the parent body breaks apart and a new individual grows from each piece. Or starfish can regrow by regeneration. If a starfish loses an arm, for whatever reason, it'll grow a new one. In budding, as you can see in the hydra, which is a freshwater cnidarian, member of the jellyfish phylum, this is the baby sprout budding up, growing off from the parent hydra. This is a form of asexual reproduction. And this shows you asexual reproduction of a sea anemone by fission. And in this situation, the splitting is almost complete. Sexual reproduction is the creation of offspring by fertilization. The egg and sperm meet, fuse their genetic contents together to form a new cell called a zygote. Egg and sperm being haploid, forming a diploid zygote, which maintains the chromosome number of the species. The sperm typically is the motile individual. It's small, usually nothing more than a head containing genetic material and a flagellum for propulsion. It's the egg with the ova, which holds has nearly all the cytoplasm and the organelles. The egg typically is not self-propelled and much larger. Sea anemones and jellyfish and sponges can reproduce both sexually and asexually. In this video, we're looking at sperm produced by a hydra. In this diagram, we have a hydra splitting by fission, two individuals. And here we have eggs produced produced by one individual. Now, depending where you are in the animal kingdom, some animals exhibit hermaphroditism, which means both parts, male and female, in the same organism. So the individual has both male and female parts. Hermaphroditism has an advantage to find a mate for animals that are solitary or less mobile. For the most part, hermaphrodites exchange gametes with other individuals and typically do not self-reproduce, although that's not impossible. Such as in earthworms. Earthworms are hermaphrodites. Fertilization. Egg meets sperm. It can occur externally, in which the eggs and sperm fertilize outside of the parent's bodies. We see this in invertebrates, fish, and yes, amphibians. Internal fertilization, where fertilization takes place inside one of the parent's bodies. The sperm is deposited inside the female, and fertilization takes place inside, as in this case of horseshoe crabs. This stimulates the release of the egg and sperm by engaging in this type of behavior. Human reproduction. Everything you wanted to know, but you were afraid to ask. Alright, humans are not hermaphrodites. Not normally. They have males and females. Both sexes have their own set of gonads. In males, it's a pair of testes. In females, it's a pair of ovaries. Alright, so the male gonads, again, is the testes, which produce the sperm. In females, the gonads are the ovaries, which produce the eggs. In addition to the gonads, there are ducts, which lead to and from, which deliver the gametes and also play a part during pregnancy and giving birth, as well as mating. Ovaries contain follicles. Each follicle will contain a future egg. And also the follicles produce estrogens. During the human reproductive cycle in females, an immature egg is ejected from the follicle about once every lunar month or 28 days. This is called ovulation. After ovulation, the remains of the follicle become a structure called the corpus luteum. And this corpus luteum secretes hormones which prepare the uterus for possible pregnancy. And again, estrogen and progesterone.
The female reproductive system consists of the ovaries, oviducts, uterus, and vagina. The ovaries produce eggs, or ova, in a process called oogenesis. Each month, an egg develops within a follicle and is released into one of the oviducts, a process called ovulation. In the oviduct, the egg may be fertilized by a sperm cell. The fertilized egg starts to develop and implants in the wall of the uterus, where it will mature.
All right. Oviducts, or also known as fallopian tubes. Oviducts are situated by the ovaries and conduct the egg from the ovary to the uterus. Fertilization typically takes place in the oviduct. And once fertilization occurs, and the fertilized egg or zygote begins embryonic development almost immediately. As that future individual develops, the egg makes it, the embryo makes its way into the uterus where it implants. The uterus is the site of pregnancy. There's a very thick muscular wall called a myometrium. It's lined with an endometrial layer. The uterus connects to the birth canal by means of the cervix. The birth canal is the vagina. This is the front view of the female reproductive system. This is an actual photograph taken with a laparoscopic and a small camera of ovulation, where the egg cell actually the ovary wall actually ruptures, releasing the egg cell. In this book, and we use the word embryo to refer to the stage of development from fertilization until recognizable body structures. So embryo is used from the time of fertilization to about the beginning of the 9th week. It's at the beginning of the 9th week body structures appear and are apparent. From the 9th week to the birth, the baby is called a fetus. Now, an ectopic pregnancy is when implantation does not occur in the uterus, but rather in the oviduct wall. This is a dangerous situation which requires surgery to correct. And this is a side view of the female reproductive tract. With the non-reproductive structures, the ones that are in italics, which are associated nearby with the reproductive tract. The female reproductive anatomy. The vulva is the external female genitalia, which consists of the labia minora and the labia majora. The clitoris is at the top of the opening. It has a rounded head covered by a small piece of skin. The head is the glans, the covering is the prepuce. The male reproductive structure, the testes, are housed outside the body in a flap of skin called the scrotum. Scrotum keeps them at a temperature below body temperature, about 4 degrees Fahrenheit below body temperature. The term testicle refers to the testes within a scrotum. In addition to the testes is a small tube called an epididymis, which stores the sperm inside the testes. The process of ejaculation is the release of sperm, where the sperm leave the epididymis, travel through the vas deferens, and then to the urethra, and then outside the male body.
The reproductive system of the human male consists of testes, various ducts and glands, and the penis. The brain, acting through hormones, controls the process of reproduction. Sperm develop in the testes in coiled tubes called seminiferous tubules. Spermatogenesis produces several hundred million sperm each day. The sperm from each testis mature and become motile in a coiled tube called the epididymis. When a man is sexually aroused, muscular contractions propel the sperm out of the epididymis and into the vas deferens, which loops over the bladder. The vas deferens joins a duct from the seminal vesicle to form the short ejaculatory duct, which with the ejaculatory duct from the other side joins the urethra. The urethra conveys the sperm out through the penis. In addition to the seminal vesicles, the prostate gland and the bulbourethral glands contribute to the seminal fluid that nourishes and protects the sperm and lubricates their path.
Alright, there are several glands associated with the male reproductive system which contribute to the production of semen. This is the fluid, which is ejaculated by the penis during ejaculation. The seminal vesicle secrete a fluid full of sugars, which nourish the sperm. The prostate gland produces an alkaline buffer which protects the sperm against the environment of the acidic vagina. The bulbourethral glands secrete mucus, which also neutralizes the acidic urine in the urethra. This is a frontal view of the male reproductive tract, including the bladder, prostate gland, bulbourethral gland, the seminal vesicle we can't see here, or the ejaculatory duct, urethra, and this is a side view of the male reproductive tract. The penis, which is normally soft and flaccid, contains cylinders. It's a spongy tissue which can fill rapidly with blood, resulting in an erection. Ejaculation is when contractions take place in these glands, which push their secretions into the urethra, along with push the sperm out of the epididymis at the same time. During ejaculation, the bladder closes off, so there's no leakage of urine during ejaculation. And then expulsion, the semen is pushed into the penis. Sperm production is controlled by the hypothalamus and pituitary.
Hormones control the production of sperm and male sex hormones by the testes. The hypothalamus, the part of the brain just above the pituitary gland, controls hormone production under the influence of other parts of the brain. The hypothalamus produces a releasing hormone that regulates the secretion of follicle-stimulating hormone, FSH, and luteinizing hormone, LH, from the anterior pituitary. FSH stimulates sperm production by the testes. LH stimulates the testes to secrete androgens, male sex hormones. Testosterone is the most important androgen secreted by the testes. Androgens shape the development of the male reproductive system. They stimulate sperm production and trigger development of secondary sex characteristics such as beard growth. Negative feedback regulates the function of the male reproductive system. An increase in testosterone and other androgens inhibits the brain and pituitary, decreasing secretion of releasing hormone and LH. This in turn reduces androgen secretion. A drop in androgens allows the brain and pituitary to stimulate the testes, maintaining optimum androgen levels.
Alright, so the brain, the hypothalamus, produces gonadotropic releasing hormone, which stimulates the anterior pituitary to release FSH and luteinizing hormone, FSH, acts upon the testes to promote spermatogenesis. Luteinizing hormone stimulates the production of testosterone, which also encourages sperm production. As those levels rise up, high levels detected in the anterior pituitary and the hypothalamus shuts off the release of and the reduction of FSH and LH, which leads to a reduction of androgens. Sperm and egg are haploid cells. They have half the chromosome number of the zygote. Now, sperm and egg develop from special diploid cells. And these special diploid cells undergo mitosis to maintain themselves and also producing the cells that will undergo meiosis to form the sperm and eggs. Big differences in gametogenesis. In other words, there's big differences between spermatogenesis in males and oogenesis in females. Spermatogenesis occurs in the male. It occurs in a network of tubes inside the testes called seminiferous tubules. Primary spermatocytes or sperm mother cells are formed by mitosis and are maintained by mitosis. But certain primary spermatocytes will undergo meiosis. So, after the first meiotic division, the offspring are called secondary spermatocytes. Secondary spermatocytes undergo meiosis too to produce spermatids, immature sperm. Spermatids undergo further development to become sperm cells. So let's look at it. So primary spermatocytes which are diploid, undergo meiosis 1 to produce secondary spermatocytes, which are haploid, which undergo meiosis 2 to produce spermatids, which develop into sperm cells. Seminiferous tubules is the location of sperm production. It's a long series of coiled tubes inside the testes. After production, the sperm are released and will make their way outside of the testes into a coiled tube called the epididymis, which is on the outside surface of the testis. Oogenesis begins before birth. So the actual process begins before a female baby is born. After birth, and with the onset of puberty, about every 28 days in a young woman, follicle stimulating hormone will stimulate one or more follicles to develop. A primary oocyte will resume meiosis. It will undergo meiosis 1. Produces a secondary oocyte, which is arrested at metaphase of meiosis 2. It is at this point ovulation takes place. And when the sperm penetrates the cell membrane, meiosis very rapidly finishes. And then the actual fertilization of the egg takes place then. So actually the female egg finishes meiosis after fertilization.
Ovulation occurs about every 28 days in a regular cycle called the ovarian cycle. Hormones coordinate the ovarian cycle with monthly changes in the uterus, termed the menstrual cycle. The start of the menstrual cycle is defined by the beginning of a woman's menstrual period. The lining of the uterus, called the endometrium, breaks down, and blood, cells, and mucus are expelled through the vagina. This process is called menstruation, and it usually lasts for the first 3 to 5 days of the menstrual cycle. The endometrium again thickens in preparation for possible pregnancy. We will combine our discussion of the ovarian and menstrual cycles to show how hormones coordinate the activities of the ovaries and the uterus. The brain controls reproduction, acting via hormones produced by the pituitary gland. While a woman is menstruating, her hypothalamus begins secreting a releasing hormone which stimulates the anterior pituitary to secrete FSH and LH into the blood. FSH, follicle stimulating hormone, stimulates the growth of an ovarian follicle in the ovary. The follicle consists of a developing egg cell surrounded by cells that nourish and protect it. The developing follicle begins secreting estrogen, a female sex hormone. Estrogen shapes development of the female reproductive system and female secondary sex characteristics such as broad hips and development of the breasts. Its immediate role is to stimulate regrowth of the lining of the uterus in preparation for possible pregnancy. Low levels of estrogen also exert negative feedback on the hypothalamus, keeping blood levels of FSH and LH relatively low. Basically what is going on here is that the brain triggers development of an egg, and the follicle containing the egg signals the uterus to prepare to support the development of an embryo. Negative feedback assures that only one follicle develops at a time. As the follicle grows, it secretes more and more estrogen. This stimulates further development and thickening of the uterine lining. Estrogen level reaches a peak just before the midpoint of the cycle. Now response of the hypothalamus and pituitary to estrogen reverses. The higher level of estrogen actually stimulates the hypothalamus to signal the pituitary to secrete a burst of FSH and LH. It is the burst of LH that triggers ovulation on about day 14 of the cycle. The ovum develops, the follicle ruptures, and the nearly mature ovum is released from the follicle and swept into the oviduct. LH, or luteinizing hormone, also causes the ruptured follicle to develop into a glandular structure called the corpus luteum. After ovulation, the corpus luteum continues to secrete estrogen and increasing amounts of a second female hormone called progesterone. Both hormones contribute to further thickening of the uterine lining in preparation for possible pregnancy. Estrogen and progesterone also exert negative feedback on the hypothalamus and pituitary, suppressing FSH and LH secretion. This keeps additional follicles from developing after ovulation. We can summarize ovulation and the following events like this. When the follicle is nearly mature, it signals the hypothalamus to trigger ovulation. The ruptured follicle, now called the corpus luteum, prevents additional follicles from developing and signals the uterus to prepare for implantation and development of an embryo. What happens next depends on whether the ovum is fertilized or not. If the egg is not fertilized, the corpus luteum has a short lifespan. Suppression of LH after ovulation causes the corpus luteum to degenerate. As the corpus luteum's output of estrogen and progesterone drops, two changes occur. The lining of the uterus begins to slough off, the beginning of the menstrual period and start of the next cycle. The drop in estrogen and progesterone also reduces feedback inhibition of the hypothalamus and pituitary. This triggers secretion of FSH and LH, which stimulates the development of a new follicle. So if fertilization doesn't occur, the corpus luteum degenerates. Without its hormones, the lining of the uterus breaks down and a new follicle starts to grow. If the egg is fertilized, it starts to develop and implants in the wall of the uterus. The embryo secretes a hormone called human chorionic gonadotropin, HCG for short. HCG functions like LH to signal the corpus luteum to continue its secretion of estrogen and progesterone. Later, the placenta takes over this function. Progesterone and estrogen maintain the lining of the uterus and suppress the development of any more follicles. In effect, the embryo signals the corpus luteum to stick around, maintaining the uterus for embryonic development and stopping the monthly cycle. Human reproduction is complicated. It depends on precise coordination of the ovaries and uterus in response to hormonal signals orchestrated by the brain.
Alright, so here's a diagram on oogenesis and the events that are going on inside a follicle at the same time. So before birth, we have the beginnings of oogenesis. So before a girl is born, she has egg mother cells or undergo meiosis, producing a primary oocyte, which is arrested at the beginning of meiosis here. And then with the onset of puberty, a follicle begins to grow, triggering the primary oocyte here to finish the first meiotic division, resulting in a polar body and a secondary oocyte which stops meiosis at metaphase of meiosis 2. At this point, ovulation occurs. With a surge of luteinizing hormone, the ovarian wall ruptures and the contents of the follicle are expelled into the oviduct. At this time, the remains of the follicle develop into the corpus luteum, which secretes estrogen and progesterone to get the uterus ready for a possible pregnancy. At this point here, the sperm penetrates this membrane, which upon the penetration of the membrane of the secondary oocyte, the egg, the secondary oocyte hurries up and finishes meiosis to produce a third polar body and a mature egg cell, which is then fertilized, resulting in a zygote. So, before a young woman is even born, there are diploid cells in the ovaries, primary oocytes. And one primary oocyte will begin meiosis 1, and then it stops. So all the eggs a girl's ever going to have are present at birth. With the onset of puberty, every 28 days, one or two follicles or more will begin completion of development. At this time, the primary oocyte will finish meiosis 1, resulting in a secondary oocyte and the first polar body. At this point, ovulation will occur. With the release of the secondary oocyte, a sperm cell penetrates the the cell membrane of the oocyte, which then speeds up and finishes meiosis 2, resulting in an egg cell being fertilized. So, the main difference between oogenesis and spermatogenesis is that spermatogenesis, a primary spermatocyte will produce 4 sperm cells. But in oogenesis, a primary ovocyte that undergoes meiosis only produces 1 functional ovum and 3 polar bodies. Polar bodies disintegrate. In males, spermatogenesis occurs from puberty to death, while oogenesis, the mitotic divisions of oogenesis are believed to be completed before birth and the production of eggs ceases with menopause. Oogenesis is only one part of a female mammal's reproductive cycle. The female reproductive cycle produces gametes, makes them available for fertilization, and prepares the body for pregnancy, which is actually two closely linked cycles: the ovarian cycle, the growth and release of female gametes, eggs, and the menstrual cycle, preparing the uterus for possible implantation of an embryo, which is under the control of hormones, which ensures that both cycles are coordinated to work together, which involves very intricate negative feedback mechanisms. There are several different hormones, which are the releasing hormone, which is secreted by the hypothalamus, regulates the secretion of luteinizing and follicle-stimulating hormone. Follicle-stimulating hormone stimulates the growth of the ovarian follicle. Luteinizing hormone stimulates follicle growth, also acts upon the secondary oocyte. It triggers ovulation, and also after ovulation the production of the corpus luteum. Estrogen, which is produced by the follicle, inhibits the pituitary, but also stimulates the hypothalamus, and also promotes the growth of the endometrium. And estrogens and progesterones produced by the corpus luteum maintain that endometrium. The reproductive cycle, the part A of the human female reproductive cycle. The hypothalamus produces a releasing hormone, which tells the anterior pituitary to release follicle-stimulating hormone and luteinizing hormone. The hypothalamus can be inhibited by a combination of estrogen and progesterone, but also can be stimulated by high levels of estrogen. Part B, follicle-stimulating hormone acts upon the follicle, stimulates the growth. Luteinizing hormone at its peak will trigger ovulation. Part C, at the same time, the first part of the menstrual cycle deals with the pre-ovulatory phase. With the onset of ovulation, the ovary goes into the post-ovulatory phase. At this point, progesterone and estrogen is secreted by the corpus luteum, which maintains the uterus for possible implantation. Part D, blood levels. No ovarian hormones. In the pre-ovulatory phase, estrogen in the blood is secreted by the follicle. Now, and at the same time, we have low levels of progesterone. A surge in luteinizing hormone triggers ovulation. In the post-ovulatory phase, progesterone and estrogen levels are high, produced by the corpus luteum, which thicken the lining of the uterus. Menstruation is the sloughing off of the endometrial lining. Low levels of estrogen trigger menstruation. Progesterone and estrogen together promote its thickening of the endometrial layer. STDs. It's sad to think that sexual activity can also bring about some serious diseases, but it does. It can transmit some very serious diseases. STDs are contagious diseases because they're spread from one person to another by sexual contact. And the thing about STDs, not all are treated equally. Some can be easily treated with medications. Others, no. With regard to viral STDs, once you get it, you got it. Now, if it's a bacteria, a protozoan, or a fungi, yes, there are medications that can treat these. But the key to treating bacterial STDs is to catch it early before any damage is done. This diagram illustrates some of the most common STDs and their cause, symptoms, treatments. The thing about viral STDs, symptoms can be treated, but cannot be cured. It used to be, once upon a time, that if you got an STD, you go to the doctor, get a shot of penicillin, you were cured. Not so anymore. Nowadays, there are STDs that will kill you. HIV, the causative agent of AIDS. Chlamydia nowadays is the most common bacterial STD. And yes, but the thing is, in males and sometimes in females, it produces no symptoms. If there's no symptoms, you don't know you have it until it's too late. Viral diseases, genital warts, herpes can be controlled but not cured, and AIDS is one of its own challenges. And because of the nature of the virus to mutate, coming up with a suitable vaccine to prevent it is many years away. And it used to be thought that AIDS was a homosexual disease, but no, that is erroneous thinking. AIDS is a human disease. There is such a thing as heterosexual infection, and 1/3 of the new AIDS infections, I meant HIV infections in the United States, is due to heterosexual contact. There is ways. Latex condoms have been shown to prevent the spread of HIV. And a study done by the World Health Organization has shown that infection rates between couples in which one partner had HIV positive, the other was not, versus couples and used latex condoms, well, the rate of infection in those couples decreased, but in the couples that did not use the condoms, it increased. So couple condom usage among participants had a lower rate of infection. Viral STDs cannot be cured, but they can be treated, but never cured. Sexual active requires responsibility. So if you're sexually active, you need to take care of yourself and see a doctor on a regular basis. STDs are more prevalent among teenagers and young adults. Best way to prevent STDs is not to do the deed, but if you're going to do it, be responsible about it. And I myself am a big believer in monogamy. Contraception is simply deliberate prevention of pregnancy, and there are several different forms with varying percentages of success. Birth control pills, if used correctly, less than 0.1% will result in, if it's used correctly, will get pregnant. A vasectomy, tubal ligation, intrauterine device, the rhythm method, withdrawal, condoms, diaphragm, spermicide. Yeah, but they left out abstinence. Abstinence means 0. And used typically, not according to directions. Principles of embryonic development. Embryonic development begins with fertilization. Haploid sperm, haploid egg fuse, form a diploid zygote. That, ladies and gentlemen, is your human egg cell surrounded by sperm, one of which will be lucky. Sperm are adapted. Their job is to swim to an egg. They have streamlined shape. They're able to move readily through any fluid. They have many mitochondria, energy powerhouses, provide enough ATP. They have a head, which contains a haploid nucleus, and it's tipped with an acrosome. Acrosome are enzymes that penetrate the egg cell membrane. The head contains the nuclear material and the acrosome. The middle piece contains the mitochondria that produce ATP that power the flagellum. This is a timeline for the events in the fertilization of a sea urchin egg. At time 1, the binding of the sperm to egg. Once the sperm penetrates the egg plasma membrane, after that penetration becomes impenetratable to other sperm. After fertilization, within 5 minutes, there's increased protein synthesis. The egg and sperm nuclei finish their fusion of genetic material and begin DNA synthesis for the first mitotic division, which typically takes place 90 minutes after the binding of egg and sperm. Alright, when the sperm touches the egg, enzymes in the acrosome begin the process of digesting their way through the egg cell membrane. There is a jelly layer, a vitelline layer on top of the egg cell plasma membrane. So these enzymes begin digesting their way through the jelly. Proteins bind to receptors in the egg cell membrane. The cell membranes of the sperm and the egg fuse. The sperm nucleus enters the ovum. Now, the vitelline layer separates. The sperm nucleus fuses with the egg nucleus, resulting in a zygote nucleus. So actually, about 20 minutes after the sperm nucleus enters the egg, their nuclei, the sperm and egg nuclei fuse, and then it starts gearing up. The zygote starts gearing up for all the growth and development that has to take place. DNA synthesis, cellular respiration begin. The first step of embryonic development is cleavage. That's a rapid series of divisions that produce more cells, similar cells, and a fluid-filled cavity called a blastocele, which in an embryo is called a blastula. Alright, 2-cell stage. Four-cell stage. A blastula. And that hollow interior is a blastocele. Now we see the early beginnings of gastrulation. Look at the bottom of the video. That structure there will end up being the lining of the future digestive tract. That opening right there at the base will be the future anus. Alright, let's take a look at this. Sperm meets egg, zygote, 2 cell, 4 cell, 8 cell, 16, 32, 64, so on. Blastula, blastocele. One side of the blastula folds in, that is gastrulation. Gastrulation initiates the beginnings of organ formation. Gastrulation is which cells migrate, the beginning of the digestive tract, and development of the basic body plans with 3 tissue layers. Ectoderm, which is on the outside, becomes the skin and nervous system. Endoderm is on the inside, forms the digestive tract. And mesoderm in the middle forms the muscle, bone, and all the other organs. At the end of cleavage, we have a blastula. This is the development of a frog gastrula. Now, gastrulation is marked by cell migration. The blastocele diminishes, and we see the beginnings of the formation of the digestive tract. This opening here, blastopore, will become the anus. And now, at the end of gastrulation, we have 3 germ layers. Ectoderm on the outside, endoderm on the inside, forming the lining of the digestive tract, and mesoderm sandwich in between, which forms the blood, bones, muscle, all the other organs. So ectoderm gives rise to the epidermis of the skin, epithelial lining mouth, rectum, sense receptors. Endoderm basically gives rise to the lining of the digestive tract, except the mouth and rectum. Lining of the respiratory system, your liver, pancreas, thyroid, parathyroids, thymus, lining of urethra, urinary bladder, reproductive system. Mesoderm is skeletal system, muscular system, circulatory system, excretory, reproductive system, the dermis of the skin, lining of the body cavity. And from those 3 layers, all the organs develop. At the same time, a notochord, a very stiff rod notochord, forms the main axis of the body. In most chordates, specifically vertebrates, it's replaced by their vertebral column. Alright, in this diagram we see the beginning of organ development in the frog. The notochord runs along the longitudinal axis. It provides support. From the ectoderm, a neural fold develops. This neural fold will give rise to the future nervous system, brain and spinal cord. So this neural fold becomes a neural tube, and that forms the brain and spinal cord. So this neural plate gives rise to the neural folds. As the embryo gets longer, elongates, structures called somites pair up and form. These somites will form the future body cavity and also develop into muscle, bone, and other tissues. At the same time, other body systems are developing as well. So this diagram shows an embryo with a completed neural tube, somites, and a coelom shown in a side view. And this is a cross section. Here, this is a side view, and this is a cross section of that side view. And here we have a tadpole. Now, the process of development depends on signals passed from neighboring cells and cell layers, so development proceeds under a coordinated process. In other words, they do what they have to do when they have to do it. Now, this mechanism by which one group of cells basically influences development of another set is called induction. Induction is how development takes place in virtually all tissues and organs. Besides induction, development occurs by changes in cell shape, the migration of cells, and timely cell death or apoptosis. This is programmed cell death, in which at specific points certain cells are going to die. This is how the fingers of a human form, the spaces between the digits, because cells die at a specific time. So this illustrates apoptosis in the developing human hand. So this is an example here. When one cell undergoes death, it will be engulfed and digested by the adjacent cell. The next step is pattern formation, where the parts of a structure emerge in their correct positions. This involves responsive genes to spatial variations of chemicals in the embryo. And by the placement of the chemicals and by the amount of chemical produced, this results in the tissues and organs being in the proper place at the proper time. Something like this. For example, in the development of limbs. Now, the development is controlled by master sets of genes called homeotic genes. And the turning on and turning off of the genes in particular sequence guide the pattern formation in embryos. We see these homeotic genes not just in mammals, but also in bacteria, and yeast, and plants. And this is one of the pieces of evidence which suggests a common ancestor for all life forms. So this is one of the key pieces of pattern formations, one of the key pieces of evidence to support evolution. Because even humans and flies have similar master gene sequences. As you can see here in the flies and in mouse embryos. Development of humans. Pregnancy or gestation is carrying of the baby within the female reproductive tract. It takes about 38 weeks in humans, or 40 weeks, depending on whose textbook you use. Human development begins with fertilization in the oviduct. Cleavage produces a blastocyst. The blastocyst is the inner cell mass that becomes the embryo. The trophoblast, which is the outer layer of cells, which implants into the uterine lining and will form part of the placenta. After this, gastrulation occurs and production organs develop. So, fertilization takes place here in the embryo, in the oviduct. So about a week after fertilization, the blastocyst and trophoblast implant into the uterus. Once implantation takes place, the beginnings of the development of the placenta. At the same time, the embryo undergoes its own development. So implantation takes place from fertilization to implantation. It takes about a week, 7 days. During the first month, 4 extra embryonic membranes will develop. The amnion, which will surround the embryo and form the amniotic sac that protects, in other words, a swimming pool for the baby, protects the baby. The yolk sac is the second, which in reptiles will store yolk, but in humans does not store yolk, but is a source of the first germ cells, reproductive cells, and blood cells. The allantois forms the umbilical cord and urinary bladder. In reptiles, it stores embryonic waste. The chorion, which in mammals contributes to the placenta, also secretes HCG, which prevents menstruation from occurring, keeps the uterine lining intact, and that's the hormone that is tested for in the home pregnancy kits. And this shows the development of in the first month from the embryonic and at the same time placental development taking place. All this takes place in the first month here. The placenta is a combination of maternal and fetal tissues. The placenta is simply the life support system for the developing baby. It provides nutrients, oxygen, removes waste products, and carbon dioxide. Pregnancy is the period of development beginning with contraception, ending at birth, and it's divided into 3 trimesters. It is the first trimester that is the most critical. This is when the most time and most radical change. And this is when the embryo is susceptible to radiation, chemicals, bumps, alcohol. Anything can upset the pregnancy or even cause an early termination or miscarriage. And typically morning sickness occurs during this time and typically lays off by the second trimester, and nobody's really sure what causes morning sickness. Just know it happens. Here's a timeline of human development and correlated with significant development events at specific times. It's during the second trimester the fetus continues to grow and has increasing human features. Changes are not as dramatic as the first. The fetus eyes are open, its teeth are forming, and its bones have begun to harden. At this time, the placenta begins to secrete progesterone. The corpus luteum stops secreting progesterone. This helps maintain the placenta, and at the same time, the placenta does not secrete HCG, and this permits the corpus luteum to degenerate. The third trimester is a time of rapid growth, and the fetus gets ready to live outside of the uterus. Babies have been born as early as 24 weeks. They can survive but require special medical care. During this time, the fetus grows, the uterus expands, and mother's abdominal organs become squeezed, causing frequent urination, digestive troubles, and backaches. The series of events that expel an infant from the uterus is what we call labor. This is under hormonal change. Estrogen makes the uterus more sensitive to oxytocin. Oxytocin and prostaglandins initiates labor, the contractions. The cervix dilates. The baby is expelled by strong uterine contractions. And then after the baby is expelled, the placenta dislodges and expelled. The induction of labor involves a positive feedback. This is in which the change triggers mechanisms that amplify the change. So oxytocin and prostaglandins cause uterine contractions. These contractions stimulate the release of more oxytocin and prostaglandins, and this causes a steady increase in contraction intensity, which climaxes in the birth of the baby. Labor occurs in 3 stages: dilation of the cervix, expulsion of the infant, and delivery of the placenta. So, a fully dilated cervix is about the length or about the width as a cigarette is long. Now, couples that are unable to conceive due to various difficulties, we have techniques to help them. Assisted reproductive technologies, eggs being harvested, fertilized, and returned to the woman's body, in vitro fertilization, in which fertilization takes place in a culture dish, and then the early embryo is implanted in the 8-cell stage. So the egg and sperm are collected, fertilization takes place, and then at the 8-cell stage we have implantation.